Good morning, students. Welcome back to my channel, Molecular Mysteries. So, in this class, let's talk about the proto oncogenes and oncogenes. So, hope you have covered the basics of cancer in the last class itself. If you have any doubt, please let me know in the comment section. In this class, let's talk about what are actually proto oncogenes and oncogenes, and how these proto oncogenes are converted into oncogenes and making it to enter into the property of cancer, like how the oncogenes, that oncogenes have the capacity to enter into a metastatic stage for cancer to happen, right? So how and what are the mutations that are making the proto-oncogenes into oncogenes? Let's discuss in this class. So hope you are all thorough with the cancer part till now, the basics and all. I will also link the basic things in the description box. If you want to go through the basics of cancer and also terms in cancer, please go through the description box once. Okay. So, what are oncogenes and proto-oncogenes? So, the proto-oncogenes are generally a normal genes, I can say, when they undergo a mutation. So, the DNA undergoes a very lot of damages, right? So, if that damage is so particular, like it's a mutation, which may have a nucleotide change, complete nucleotide change or deletions. Anyway, I'll also talk about the type of mutations that are making the DNA defects in the DNA. Okay, I'll also talk in the next coming class. So in this class, now let's talk about proto-oncogenes, which have the normal gene capacity. They're generally normal genes which helps for the cell cycle mechanism. But these proto-oncogenes, when they undergo a mutation, they get converted into a oncogene. Hence, the oncogenes would get the property of undergoing angiogenesis, also the property of undergoing a metastasis. Okay, so I told you these are the two differences between the proto-oncogenes and the oncogenes. So oncogenes are mutated and are overexpressed proto-oncogenes, which have the ability to promote the cancer, and they can be in a dominant stage. And generally the proto-oncogenes are generally normal genes. Only when it undergoes mutation, they get converted into an oncogene. Okay, now let's see how these are converted. So this is the main important chart. So I told you the proto-oncogenes are generally the normal genes which help for the protein mechanism and obviously the normal cell cycle mechanism. That is normal for all the genes to happen. So right, all the protein synthesis should happen. A chain of amino acids should be produced, and this protein synthesis helps in the proper amino acid sequence with the help of the growth factors. Right. So this is how the proto-oncogenes would normally participate in all the cell cycle mechanisms. But when these proto-oncogenes are or like getting under the mutations, it could be point mutations, it could be translocations, gene amplifications. Okay, I will also discuss that anyway. So it could be missense mutations, right? It can be gene translocations also. So these are all the types of mutations which could convert the proto-oncogenes into oncogenes. And once the cell has converted itself as an oncogene, it starts producing the proteins called oncoproteins, which are totally different from your normal proteins. And these oncoproteins doesn't need any growth factors. Already I've told you, one of the important property of the cancer cell is doesn't require the growth factors. The oncoproteins, the cell itself produce the oncoproteins which are helpful for the cell to undergo more and more ungrowth undivided cell proliferation, right? If you want to see, I can also attach it. So one of the important properties of the cancer being is that it doesn't need any growth factors. So normal cells need growth factors. The oncoproteins by itself help the cells to produce more and more growth factors in a wrong way. Thus it leads to angiogenesis, which is a production of more and more blood supply to the growing cell, undeveloped or unregulated cell proliferation, right? And leading to the malignant condition. So that is how the oncogenes convert in themselves into malignancy. Okay, so this is the main important short topic of conversion of proto-oncogenes into oncogenes. Now let's see what are the steps. So generally, know that cell cycle regulation, generally these are all are happening in the proto-oncogenes. When I told you proto-oncogenes, these are all the normal genes. So normally what happens, you have got a receptor for every growth factor, right? Each and every cell has got a receptor. So but first of all, the growth factor should come and sit onto the receptor, then only the activation of growth factor would happen, which leads to the signal transduction pathways, right? Thus the transcription process happens. Once transcription process happens, you have the translation happening, you have the proteins releasing, and you have your cell cycle progression. This is how the process of proto-oncogenes would happen in a normal cell. So when it is getting hit by a mutation, that leads to the oncogene. So now let's see how the mutations happen. So I told you the mutations that are converting the proto-oncogenes into oncogenes are point mutations, gene amplification, chromosomal translocation, and viral integration. So anyways, I will discuss that in the next class. So in a short, I can say you the point mutations are you all know that the genetic code, right? So all the four nucleotides, adenine, guanine, cytosine and thymine, these are the four important nucleotides. Based on the arrangement of these four nucleotides, you have your genetic code made, right? So what happens? Three nucleotides code for one amino acid. If you are well aware of the transcription and translation process, you will be knowing this process. So okay, the three nucleotides at a stretch will code for one amino acid, and if any substitution of the nucleotide or deletion of a nucleotide or addition of one more nucleotide happens, this leads to a point mutation, which is a permanent damage for the DNA, right? So that is what you call the point mutation. So gene amplification, right? Like if you have codon, like coding for a valine amino acid, okay, but what happens if you have more and more expression of that particular codon more times, more than the required condition? If you require only a little amount, but it's been required more and more, and it keeps producing the same gene more times, gene amplification more than required, it also leads to the gene amplification, chromosomal translocation, and viral integrations, right? You have direct link between these also. So when you come across the genes like hepatitis B, so hepatitis B directly causes a liver cancer, right? So there are many genes like gene defects in BRCA gene B1 and BRCA gene 2, which causes the breast cancer and the ovarian cancer in ladies. So these are all defects would happen in the gene. And apart from that, you also have human papilloma virus, HPV virus, which directly gets linked to your cervical cancer in ladies. So the viral integration is also making the proto-oncogenes converted into the oncogenes, right? Right. Then what I told you, these are all missense mutations. These are underpoint mutations. You have your missense mutations and nonsense mutations. So you all know when you go across the transcription process, sorry, translation process, where happens, the ribosome keeps moving on the mRNA, and it has to produce correct codon coming from the transfer RNA, and that is how a chain of amino acids are made. So when there are nonsense mutations, I told you a single nucleotide change can introduce a premature codon. So you have UAA, UAG, and UGA, which are the stop codons, right? When this comes, the translation has to stop. When, for example, if there is a mutation happening in the mRNA area where the UAA, UAG occur a little moves further than our normal location, obviously the whole translation process would stop because of that, and thus it may lead to a point mutation. So these are all the mutations that may change the proto-oncogene into your oncogene, right? Now let's see gene amplification. I told you under the gene amplification, you have gene duplication also. The proto-oncogene is duplicated, leading to the overexpression of the proteins, and the gene amplification. The proto-oncogene is amplified, resulting in the multiple copies of the genes and also the subsequent overexpression. So these are all amplification is increase in number, right? So gene duplication and gene amplification would also cause a mutation in the proto-oncogenes. Hope you are thoroughly following my channel, sorry, following my class. And a chromosomal translocation. So these are the important mutations like balanced translocation and unbalanced translocation. So what is it? A reciprocal change of genetic material between the chromosomes can bring proto-oncogenes into an oncogenes. Under this, you have a promoter also. So there is a promoter for every translation process to happen. You need a promoter, right? So if there is a strong promoter, that is okay, but if there is any change in the reciprocal, which is a reciprocal one, this is called a balanced one, translocation, a reciprocal exchange of genetic material between the two chromosomes, okay, can bring the proto-oncogenes under the control of a strong promoter, obviously, right? If you have two chromosomes, if there is a location, there's a complete change of one part of your chromosome getting exchanged with the other part of your chromosome, obviously it gets control of under the strong promoter, which is a non reciprocal change. And you have unbalanced translocation also, leading to even more bigger one, which is a non-reciprocal exchange of genetic material leading to the loss or gain of genetic material, right? So unbalanced, either it could be it added more amount of chromosome, or it could be a deletion or part of your chromosome getting deleted. So both get the translocations are the examples for your chromosomal translocation, right? And now let's see viral integration. So I told you human papillomavirus causing the cervical cancer, and you have your liver cancer caused due to hepatitis B virus, right? Apart from that, you have the insertional mutations like the integration of your viral DNA into the host genome, right? It is also coming under the example of the viral integration, making the proto-oncogene into a oncogene under a strong promoter. And these mutations can occur spontaneously, which could be due to ultraviolet radiations, or it could be due to chemicals. So if you have any doubt, please let me know in the next class. So in the next class, I will talk about the DNA defects in the DNA repair mechanism, right? We should be well aware of what are the DNA repair mechanisms. Whenever the DNA gets under the if it gets affected by any area, like it could be chemicals or it could be a small threat, it has to repair by itself. When that doesn't happen properly, then only it leads to the cancer. So hope I'm thorough with the class till now. So I will also add the main important descriptions in the description box. Please go through it, and thank you and have a nice day.